introduced the structure of the kidney, where we were able to see the different structures, the different layers of the kidney known as the renal parenchyma, and we were able to see the nephron as the functional unit of the kidney, and the different types of cells and the different types of different parts of the nephron, and how each part is involved in urine formation. Now today we are going to talk about the physiology, the science behind the urine formation. So you have to first understand the anatomy before you understand how actually it works. Now the process of urine formation is divided into four processes or 4 stages. 4 stages are involved up to when you pass out the urine, and all the 4 take place in the functional unit, which is a nephron. And the first one is the ultrafiltration. You can call it the glomerular filtration, and it takes place in the renal capsule, or you can call it the Bowman's capsule, but to be specific, in the glomerulus. Okay, that's where this process takes place. Then after forming the glomerular filtrate, then you go to the second process, which is selective reabsorption, or you can call it tubular absorption, and it takes place throughout the entire tubes, right from the proximal convoluted tubule up to the distal convoluted tubule. Now another process that is involved in urine production is tubular secretion, which involves addition, okay, or secretion of materials that have not been filtered in blood. For example, wastes, hydrogen ions, ammonia that is produced within the cells, they are actively secreted. They are secreted into the renal filtrate. So we're also going to talk about it. And finally, there's water absorption. Okay, water must be absorbed, because yes, as you're forming the filtrate, actually you form the filtrate formed, almost 95% of water is reabsorbed back. And you're going to talk about the process, the effect, and how the nephron performs that. And then we talk about the countercurrent multiplier effect, and how actually the kidney is able to perform that, and how actually this effect plays an important part as far as urine formation is concerned. So welcome guys, and let's check together and see how actually this urine is produced in our kidneys. Let's start with process number 1, ultrafiltration. Now this is filtration, like, you know, filtering, to filter. You have, maybe you're making some juice, and after squeezing the banana, after putting it in a blender, and you create that mixture, you filter out the juice and the residues remain. So that's filtration. But now this is ultrafiltration, which takes place due to the influence of high hydrostatic pressure. Don't just say pressure, because there are different types of pressure, but this one is specifically hydrostatic pressure. So the hydrostatic pressure that is created within this glomerulus, it's the one that forces materials that forces small molecules to be filtered out, to leave blood and form this glomerular filtrate. And so how is that hydrostatic pressure built up or created within this glomerulus? Now it is simple. From this drawing, from this diagram, it's not a drawing, from this diagram, this is basically a renal capsule, okay, or a Bowman's capsule, which is made up of a swelling, which is the capsule, and then the glomerulus. You can see that we have the afferent arteriole, which brings in blood into this Bowman's capsule, and then you have the efferent arteriole, which takes away blood from the glomerulus. And then the afferent arteriole, it branches into a network of numerous blood capillaries. It's a dense network of capillaries. So now, if you had to compare the diameters of the two, the renal artery, so the afferent arteriole and the efferent arteriole, if you had to compare the diameters, the afferent arteriole has relatively a larger diameter than the efferent arterioles. Meaning, this one brings in large volumes of blood than the blood that is leaving. And remember the pumping action of the heart. The heart always pumps, keeps on pumping. So there is too much pressure. Blood that is flowing through this glomerulus, it flows under high pressure. And mind you, you are bringing in large volumes of blood. But as if that's not enough, when blood reaches here, it is going to branch into a dense network, okay? Dense network of capillaries that will all collect up into one large arteriole, which is the efferent arteriole. Now you're bringing blood in large volumes, large quantities, but this blood has to squeeze through this dense network of capillaries. So there is too much resistance that is created in the process. But due to the pumping action of the heart, you keep pumping, but there is too much resistance. So as a result, you're going to create a high hydrostatic pressure that is going to force small molecules to diffuse across or to diffuse out of the capillary lumen or the lumen of this capillary through the epithelial wall, the endothelium, and through this epithelial layer that surrounds this capillary, these podocytes, and makes it into the glomerulus. So that is the hydrostatic pressure, okay? And it is identical to the pressure that is involved during formation of the tissue fluid, okay? Yes. So it's because of that pressure that you're going to force. It's force by force. You forcefully squeeze materials or push materials out of the lumen of these capillaries into the glomerulus. But not all materials, by the way. Not all materials. But materials that are made up of small molecules, they are the ones that can squeeze through those tiny pores. Now, the other time we talked about the structure of a podocyte. We talked about a podocyte. And we say that the podocyte has the finger-like structures. So this is your podocyte. And you can see how these finger-like structures, they do surround this wall of the capillary. Okay? So this one is a capillary. This from here, this is the basement membrane. of the epithelial cells, and then this is the membrane of this podocyte. Now these are the foot-like structure. So they form slits which do allow small molecules to penetrate through. But the small molecules, they do penetrate under the influence of large pressure, of a high hydrostatic pressure that is created. But now for large molecules, such as blood cells, platelets, and large-sized proteins, those plasma proteins such as albumin and other plasma proteins, they cannot filter out. They cannot filter out. So they cannot constitute our glomerular filtrate. They will stay locked up regardless of a high hydrostatic pressure created. But the rest of the small molecules, talk about glucose, talk about water, talk about vitamins, talk about amino acids, talk about ions, the inorganic ions, urea, uric acid, depending, they will be forcefully filtered out and they form what we call the glomerular filtrate. Also guys, yes, so the movement across these pores is passive, doesn't involve any energy, but it is due to the influence of too much pressure, too much force that is created, okay, that is going to force these materials to filter out.